Hello biology. Today we're going to talk about cell growth and division. So this is going to be a lecture over chapter 10.1. To start us off, let's see. We're going to talk about the reasons why a cell has to divide. And the best scenario that I can think of for this first one is putting ourselves in a library. Say our school has a decent sized library. Let's say though then Laporte is going to come and join our school, and then we're also going to have Portage join our school, but the library stays the same size. Well, if the library were to stay the same size, but we triple the amount of students, the demands on our books are going to be very high. So we increase that demand. We can't keep up with that demand and supply more books. So the same can be said for the DNA. DNA, if a cell grows larger and larger, there's going to be more organelles. And those organelles have to go through their certain jobs, and they need instructions from certain genes. So if it grows bigger, the genetic library, or the demand becomes too great on that genetic library, similar to a library if our school were to grow. Another reason why is the material exchange can't keep up. So as a cell grows bigger, demand for transport across the membrane is too great. So if we go, the ability to transport oxygen, food, waste across the membrane depends on the surface area. So it depends on how large the cell membrane is. The need for these things depends on the amount of organelles and stuff that we have inside. So that's going to be the cell volume. So as a cell grows, these don't increase at the same rate, and we can see that by looking at this diagram here. So if we look at this smaller cell here, we can see that it has a ratio of a 6:1, so our surface area to volume ratio. We look at and we increase our size, or we double our size of that cell, and now we have a 3:1 ratio. Okay, and then so we have then a 2:1 ratio. What you see here is we see that the surface area is not increasing as fast as our volume, because our 3x3 cube here is now at a 2:1 ratio when it used to be, when it was smaller, a 6:1 ratio. So surface area just can't keep up. The things that need to come in and out, we are saturating all of our proteins that allow things in and out, and it's basically like a highway and a traffic zone. So if you think about Chicago or traffic during rush hour, that's pretty much what it would be like if a cell were to just increase its size. The streets would not be able to keep up with the demand of the city. So as a cell gets bigger, it needs more food and oxygen, but can't transport it fast enough or in big enough quantity. Hello biology, today we're going to go over the cell cycle. It's chapter 10.2. And to go through the cycle, there are a few images I think that help us more than really writing down a lot of words. So the first one we want to take a look at is maybe just drawing a pie chart. So when we draw a pie chart, the cell cycle can be basically split up into different phases, and those different phases, similar to you guys going through phases in your life from you're an infant to a toddler and so forth, your cells are going to go through those phases as well. So to start us off, we're going to start our cell cycle in interphase. Now interphase is actually going to take up most of the cell's life. So we're going to draw that by showing our arrows. So this right here is interphase. So in interphase, we can actually then divide it into 3 other phases. So we have the first one, which is G1. We then have the second one, which is S phase, and then our third one is G2. Now in G1 phase, what we're going to see is the cell is going to just perform its normal function. So it's just going to live the life of the cell. In S phase, we call that S phase because of the synthesis phase. The cell, before it can even think about dividing, it has to copy its DNA. Because imagine if we just had a cell that didn't copy its DNA. So we can talk about a human cell. We would have our 46 chromosomes. And then if it went through mitosis without our replicating our DNA, we would have 23 chromosomes here and only 23 chromosomes here. Now the problem with that is, well, we're missing half of the chromosomes needed for the cell to perform its normal function. So they need both sets, one set from mom, one set from dad, in each new cell. So the S phase is going to be an important phase because that's where we're going to replicate our DNA. All right, after we replicate DNA, we're going to go into G2 phase. So G2 phase is going to be where the cell will prep itself for cell division. So we have prepare for mitosis. All right, so then we go into our division. So right here, we would consider this cell division, this part here. And cell division is termed M phase. And the way that I like to remember that it's called M phase is because of mitosis. So cell division will start off with mitosis. So we have mitosis branching off into a few different phases. So we start off with prophase. And I'm just going to do like this, prophase. Then we have metaphase, anaphase, and then telophase. Okay, so this right there, that is just mitosis. Now remember, mitosis is just when the nucleus divides. So we have mitosis, and we want to remember that it's just nuclear division. So nuclear division. After mitosis, and we have two brand new nuclei, we have to then go through cytokinesis, where we split up the cytoplasm. So this last one would be cytokinesis, and we'll just start it off with cytokinesis. So our nucleus, we have two new nuclei. Now we have to divide everything else in the cell evenly so that our two new daughter cells we formed have the organelles they need, and we can then go from there. Alright, so let's go ahead and take a look at mitosis at a closer glance. I'm going to go ahead and erase all this. If you need to go back and you want to study that again, go ahead and just rewind. But let's look at mitosis. Alright, so we know at the beginning, no matter what, a human cell will have 46 chromosomes. At the end of mitosis, we will have two new daughter cells that will also have 46 chromosomes. So we have complete or exact replica of the original cell. To start us off, we'll go ahead and draw our cell, draw that in pink. And we'll just have it what it would look like in interphase. Now remember, if you're doing the hand mitosis and our fingers represent chromosomes, for interphase, I say that you should represent your chromosomes all bundled up, because when it's all bundled up, then it kind of shows that our chromosomes are not like tightly wound together. And when they're not tightly wound together, they're in a form called chromatin. So let's go ahead and make sure we draw the nucleus. And in interphase, so I call this our spaghetti DNA. And interphase, our G1, our cell is going to go throughout its normal functions. Then in S phase, we're going to replicate that DNA. So we're going to add even more. So we always start with G1. Then in S phase, we go and we replicate that. So we even increase the amount of DNA that we would see in here. So then that's S. And then we go into G2. So remember, G2 then is just going to prep for mitosis. It's going to create more cell organelles so that when we do have mitosis, it's prepared to divide. After interphase, we start M phase, and M phase starts with the first phase of mitosis. And to kind of make this easier on our lives, we will start seeing that our chromosomes are now or chromatin is now in the form of chromosomes. And to kind of show you how this looks, we would have replicated DNA. So this is showing you these two are replicated strands. So this contains the same instructions as this. It's just our replicated DNA. That's what we got from Dad. One chromosome we got from Dad. And here is its homologous pair. So this one we got from Mom. And then we have another one, another replicated chromosome that we got from Dad, and we have another one then that we got from Mom. So what you can see here is we have 4 total chromosomes. We have 2 from Mom and we have 2 from Dad. That means, and I know we haven't talked about this number a lot, which is fine, we're going to get into it more next chapter, but their 2n number, their diploid number, is going to be, for this cell, would be 4. Because we see 1, 2, 3, 4 chromosomes. Okay, so don't be confused that we see this replicated pair. Alright, so then during the first phase of mitosis, we had prophase. So prophase, if we remember we're doing our hand mitosis, we can see individual chromosomes now. So that's why I have us looking like this. So we're seeing our fingers, we see individual chromosomes. Eventually in prophase, our nuclear membrane will eventually disappear. So we'll make that disappear. Okay. And then we have our centrioles. Okay, they will start to go and they will start to form their spindle fibers, and they'll move to the opposite ends of the pole. Okay, and so these are the spindles, okay, that pull chromosomes apart eventually, or chromatid sisters apart. And that's pretty much prophase. So if you can still see a nuclear membrane and you can see your chromosomes first appear, you know that you are in prophase. Alright, so then the next phase, we have prophase, and then we go through metaphase, so our chromosomes are lined up in the middle. For this one, what we would see is, let's see, go ahead and delete this here. So we are then on our metaphase. Our centrioles are going to be on opposite poles of each other. Spindles coming out of them. Okay, so we can show the spindles coming out. And our chromosomes are going to line up in the middle. Now, chromosomes don't have to line up in any order, so we're just going to put up this one here. We'll put the longer one from Mom right here. And then we can do the longer one from Dad here. And then we have this shorter one here. So they don't line up in any order. It's all random how they will actually just situate themselves. So in metaphase, we can see that all of our chromosomes are lined up in the middle. Well, then we have to detach those sister chromatids. So this is one chromosome. Each side of these contain the exact same information, but we call them, when we start separating them, chromatids. Okay? So the next phase we have, when we're pulling apart our fingers, when that A can help us signify our anaphase. So here's anaphase. So in anaphase, those spindle fibers are going to pull, and we're going to start seeing these move apart. I wish I had something to hold it. So these are going to just start moving to opposite ends. Okay? And then as they start moving apart, once they are finally apart, and then they're going to start to unwind, nuclear membrane will then reappear. We have telophase. So we would have all of these go back, basically, into chromatin. 2 new nuclei will be present. And then we'll go into our cytokinesis. So remember that mitosis stops with telophase. Okay, so remember, mitosis is nuclear division. Okay, cell division includes mitosis and cytokinesis. So in an animal cell, an animal will start to burrow in because they have that ability. They don't have the cell wall that makes it really hard for this cell to start to pinch in. And eventually it'll pinch in until there are two brand new cells available. Now in a plant cell it's a little bit different. Instead of pinching in, they have that cell wall. So it's really constricted. So let's say this is a plant cell. The cell wall is a very rigid structure, so it can't pinch in. So what has to happen when we have our two new nuclei, a cell plate is going to form, and the plate is going to move from inside out. Animals, they move from outward to the inside during cytokinesis. But for the most part, that is our chapter 10.2. So I hope this helped.